This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM.
This is TWiM, This Week in Microbiology, the podcast that explores unseen life on Earth. This is episode 312, recorded on May 30th, 2024. I'm Michael Schmidt, coming to you from Charleston, South Carolina, and joining us today from Ann Arbor, Michigan is Michele Swanson. Welcome, Michele.
Hello. Delighted to be with you all.
And also joining us today from St. Louis, Missouri is Petra Levin. Welcome, Petra.
The alternate pronunciation for Missouri. It's great to be here on this cicada-filled, beautiful day in June.
Oh, are they out, Petra?
Oh, they are out. They are outside my window, and we have brood 19 with their red eyes.
Yeah. And so you crunch when you're walking? Does it crunch?
They're not too bad right where we are. They're really interesting, though, because some older trees, you can see them swarming above it. And then places that have had like new developments or even not, the trees are under 20 years old, really don't have many. And so some areas have a lot and some areas don't. And because they burrow under the trees, they're amazing. Yeah. They count cycles by the way that these are 13-year cicadas that we have. We only have one brood, that the sap comes through and they can count up to 13. But these guys are derived. Sorry to nerd out on this, but they're derived from the 17-year cicadas. They're sort of a derivative brood. And those guys count 17 and they're not sure how they count at all, but the fact they count differently and they're really curious. I think up in Michigan, maybe you get both broods together.
We're not getting any of it up in Ann Arbor and north of here.
Okay. Because some places, maybe the mid-Atlantic, they have both broods together, which means they've doubled the amount the first time since like the early 19th century. And I think they might, mate and what number of years they'll get out. I mean, this is in addition. We always have cicadas here a little later. They're green. They look very different. But dogs love them. There's a lot of dogs who've eaten too many of them.
I'm a little jealous to miss this major biological event, but I'd rather hold out for northern lights than cicadas.
I think a northern light, my miniature schnauzer would disagree with you. She's pretty into them, but I agree. Less messy.
And finally, our colleague, if you will, our podfather, Vincent Racaniello, is away this week doing what he loves, recording a science podcast with friends. But unfortunately, he's in a different time zone. He's 6 hours ahead of us in Sweden. So rather than disturb his dinner, Michele and Petra and I are going to bring you two papers this week that we hope you'll find as fascinating as we do. I have the honor of presenting our snippet today, which is another paper from our colleagues to the north, from Canada, specifically McMaster University in Hamilton, in the province of Ontario, where the authors are in the departments of biochemistry and biomedical sciences, medicine, the Farncombe Family Digestive Health Research Institute, and the Michael G. DeGroot Institute for Infectious Disease Research. And the authors are Alani, Fatima, Nair, Mayall, and Hines. And the paper that they authored that appears in this month's MBio is Temperate Phage-antibiotic Synergy across antibiotic classes reveals new mechanism for preventing lysogeny. And it's open access, so you all should be able to follow along. As some of you may know, I worked on phage therapy for the better part of 10 years, about 20 years ago. So when I spotted the title, I was sufficiently intrigued. And then when I read the paper, I was amazed. And the data, again, breathed some new life into the infection therapy approach made famous in 1925 in the Pulitzer Prize-winning novel by Sinclair Lewis in his book Aerosmith, which, spoiler alert, if you have not read the book, follows the life of Martin Aerosmith, a young doctor who struggles to balance his desire for scientific discovery with the practical realities of medical practice. So it's part satire and really part true story that many of us translational scientists face when our well-intentioned or well-reasoned scientific ideals or ideas don't quite go our way. So here's the story. Temperate phages, or should I offer the cis-acting elements contained within the genetic material of viruses of bacteria, are our planet's original quarterbacks. Here, evolution has optimized the genetics of phage for fitness, with its outcome being to facilitate phage as being the largest of all the biological entities on our planet, with a population density of approximately 10 to the 31. That's bigger than a mole. So the cis-acting elements selected over countless generations functions immediately upon its abiologic injection into a new host. In this case, in today's paper, it's going to be DNA coming from this temperate tailed phage into E. coli. And anyone who has ever read Mark Potashny's 1986 seminal 154-page book on phage lambda entitled The Genetic Switch will immediately appreciate the significance of the paper, whereas the phage begin to expose themselves to the intracellular milieu of its new host, this cis-acting phage element assesses not only the state of the host, asking, if you will, if things are good. The cis-acting quarterback then calls the play and then will integrate its nucleic acid into the genome of the host or not, where it hopes to live long and prosper for many successive generations. However, as any quarterback who has ever been sacked can appreciate, occasionally something goes wrong with that front line, but like all good scrambling quarterbacks, the phage quarterback quickly adapts, leaves the pocket, vacates its happy home in the genome of the host, but before it accomplishes one hell of a run that we can all appreciate as the lytic infection, where it makes between 100 to 300 progeny phage and literally blows up the host, ensuring an opportunity for another generation of phage that is then going to call the option play, whether or not to integrate or whether or not to kill the host. So with that as background, and apologize for the football metaphor, but it really helps us understand the really elegant biology that these authors entertained in this paper. So what the authors are trying to go about trying to decipher is this initial option behavior of calling whether they're going to effectively go into the host or not. And the way they did this is quite clever. What they learned is they could bias the call of the molecular quarterback or that cis-acting element by adding antibiotics at the same time they introduce the bacterium to the phage. For some antibiotics can lead to the activation of the host repair or Rec-A-dependent SOS system. Now, the SOS system is highly dependent on the protease Rec-A, which is a 38 kilodalton protein that is essential for repair and maintenance of DNA, literally in everything. In fact, a Rec-A structural and functional homolog has been found in every species in which one has been seriously sought and serves as an archetype for this class of homologous DNA repair proteins, where it has multiple activities all related to DNA repair. And in the bacterial SOS response, Rec-A also has a different function. It functions as a co-protease that cleaves autocatalytically the LEX-A repressor as well as the lambda repressor, the DNA protein binding molecule that keeps the cis-acting element in the chromosome, keeping the lytic genes in their off state.
It's so amazing that it does this. This is like
the
ultimate like ejection seat, because for the phage, they have to have, the one thing they need is accurate DNA replication. And that they've essentially co-opted the DNA repair system and the same catalytic mechanism, it's so cool to get out of this being lysogenic is very cool. We teach this in class, because it's so neat.
And Mark Potashny's book, The Genetic Switch, at 156 pages, is eminently readable. It's in its 3rd edition. You can buy an inexpensive paperback in any number of websites. I put into the show notes the Cold Spring Harbor reference. It's up to the 3rd edition that was published now 20 years ago in 2004.
Yeah, I think the original edition is the best because it's the most tight, and his prose is so good, and it doesn't have anything extra. It's really about the bacterial system. Yeah.
And the way I looked at this, if you will, cry havoc and let slip the phages of healing when in the presence of an antibiotic that lets loose this quarterback to call for a lytic infection simply from LEC-A, cleaving this repressor that's keeping the dogs of war at bay simply by turning the lytic expression profile from off to on. And as background, the authors rightly offer that there has been a recent demonstration of this synergy of adding a temperate phage to an infection and then adding the antibiotic ciprofloxacin, which is a fluoroquinolone class of antibiotics that principally inhibits DNA synthesis through its interaction with DNA gyrase. And as many of you know, DNA gyrase is involved in the winding and rewinding of DNA. And if it's not done properly, and that's what the fluoroquinolone class of antimicrobials do, it interferes with that winding, the DNA is damaged. And the authors thought that this observation suggested a scalable approach to exploiting temperate phages for therapy. They even coined a term and created an abbreviation. So they're serious. They abbreviated that TPAS, T and then PAS, and this was termed temperate phage antibiotic synergy, which specifically interacts with this lysis lysogeny decision matrix. So what the authors did to determine whether this would hold true across antibiotics is they challenged a particular strain of E. coli with the phage HK97, which is a tailed phage like lambda, and they used a set of 13 different antibiotics spanning 7 classes, where the class of antibiotic describes principally its mode of action. And as expected, given the conserved induction pathway where the phage opt for lytic expression profiles in the presence of an antibiotic that activates or kills that repressor molecule, the lytic infection simply goes off simply because it cannot turn off the lytic gene expression. And so the genes just get expressed. The polymerase finds it because there's no repressor sitting on the gene. Now, this observed synergy occurs with classes of drugs known to induce an SOS response. So things like a sulfur drug that we commonly use for urinary tract infections. Sulfa drugs, recall, inhibit thymine synthesis by targeting dihydrofolate reductase, which inhibits folic acid, leading to DNA damage. Quinolones, again, we've already explained those. DNA gyrase is the one that it targets in gram-negatives. Topoisomerase 4 is more common in the gram-positives. And mitomycin C, an antimicrobial that we don't often hear about, it induces single-strand breakage, which is, I always view mitomycin C as the catnip to REC-A because it creates that single-stranded break, and single-stranded DNA is a known activator of REC-A. So then REC-A's protease is tuned way up and all the lysogens come out to play. So interestingly, though, the authors offer that some beta-lactams exhibit synergy, but here they will show that the mechanism has nothing to do with the genetic switch letting loose the lytic expression profile, but rather a traditional phage-antibiotic synergy with no effect on this lysis-lysogeny decision matrix. But like most times when the dogs of war are released, they curiously observed a potent synergy with antibiotics not known to induce the SOS response. And surprisingly, those were the protein synthetic inhibitors like gentamicin, kanamycin, tetracycline, and azithromycin. In fact, from the resulting synergy, and this is what really blew me away, they observed an 18-fold reduction in the effective minimum inhibitory concentration of the antibiotic gentamicin. And they saw complete eradication of the bacteria when administered at the suboptimal 8-fold lower dose. And then they looked to see whether or not that increased the frequency of lysogens emerging from the combined challenge. They did the appropriate controls. And the lysogens that did result exhibit no increased sensitivity to the antibiotic. Now, all of this was beautifully presented by these authors, and they follow a similar format where they begin to tease apart the mechanism of how all this is working. And they accomplish this via a checkerboard assay, which is also called a sinograph, which you can visualize and easily interpret in their figures. But if you don't have the paper in front of you as you're jogging, on the y-axis, you have the MOI, or the multiplicity infection, or the phage to bacteria ratio. Now, the reason this is important, the higher the number of phage, the greater the likelihood that the phage will find a bacterium into which it can inject its DNA. And if you have too many phage, you get too many injections, and you get lysis without any productive infection. On the x-axis, you have the concentration of the drug. And on the y prime axis, you have the percent area under the curve, where they are comparing it to an uninfected phage control. And they're looking at lysis to the untreated host. And they go through a beautiful series of figures, but one of their critical experiments, and perhaps their most exciting data, was not that they saw temperate phage synergy, because they did, with and without antibiotics that could induce the SOS response, but they also saw non-SOS antibiotics inducing this antibiotic synergy, which was beautifully summarized in their summary figure of figure 6. Here, the antibiotics that activate the SOS response flip the switch through SOS, but what was most provocative to me was the observation that the protein synthetic inhibitors also synergized with the temperate phage, but did so by blocking entry to lysogeny. They effectively clubbed the quarterback in the head so it couldn't make any decision. And the DNA, or the cis-acting element, had no way of knowing whether to integrate, so it just went on to its lytic form, and then it effectively killed the host. So that is effectively how the protein synthetic guys were inhibiting the process. They, of course, have to do some more work deciphering the precise pathway behind how the protein synthetics are going about accomplishing this. But then finally, we had the odd ducks, the beta-lactam class of antimicrobials, going after cell wall biosynthesis. And the data that they offered support that the synergy they saw with the beta-lactams was somehow independent of the lysis-lysogeny decision. So summing up, the temperate phage, HK97, when used in vitro, can lower the effective MIC of antibiotics belonging to 7 different drug classes, raising the next question of whether or not this could be used in clinic, where temperate phages might act as a safety net, where when given in concert with someone who's infected with a pathogen, in concert with antibiotics, might help the infected individual clear an infection, as we see these ever-increasing MIC thresholds where we just can't give enough drug without crushing one's kidneys. But the one But the one thing that gap smacked me up my side my head was that they uncovered a method to block lysogeny, namely protein synthetic inhibitors, which now offers us Aerosmith aficionados new insight into how this phage quarterback is actually reading the line, the defensive line, and clearly more work needs to be done. So that's it in a nutshell. I'll leave you guys to sweat the details as you go through this fine paper. But I know Michele and Petra have a doozy of a paper to bring to us, and it's entitled Salmonella typhimurium expression in the inflamed murine gut is dependent on aspartate derived reactive oxygen species mediated microbiota lysis. So Michele and Petra, take it away.
Before we get to this paper, I wanted to make a couple comments on this one because I think it's really fascinating. First of all, they're using bactericidal protein synthesis inhibitors, not static ones. So I'm curious what the static ones do. And I'm wondering, since basically the reason lysogeny sometimes happens is because you just happen in a very small number to get enough of the repressor protein over this other protein called CRO. And so repressor wins. But if you just alter protein synthesis, I'm wondering if you just shift the balance.
That's my suspicion.
The easy explanation. And the beta-lactams are damaging the cell envelope, and that's probably somehow
bad news. They blow up.
All of these, though, eventually lead to reactive oxygen species, which will cause DNA damage. So there's also that possibility that even though they're not technically damaging DNA, that there's collateral DNA damage. So that's also something to consider.
The reactive oxygen species, I think, are what will ultimately set SOS off.
Right. And all of them that are cidal will at some point, I think, lead to some ROS. So I'm curious. Anyways, sorry. Thank you. I didn't want to
please.
The transition, but I just wanted to think about the different things. There's so many different things at play here. It's cool. And it's also cool because you might be able to get some, we don't always know why some protein synthesis inhibitors are bactericidal, kill bacteria, and some are static. It's not clear why. And so maybe this can also help get at some of these really mechanistic questions about why antibiotics actually work.
And which is why I dropped into the show notes a bunch of interesting references. I dropped the reference to Patashny's book and a few other things that I think our listeners will find interesting.
And the authors did point out that, yes, it may have potential, therapeutic potential, but also that it can really help experimentalists who are studying these questions in the phage bacterial interactions and interactions with antibiotics. So on to the next paper. This was published. It's open access in Cell Host Microbe. It'll formally be in the June 12th issue. And it is by groups from Vanderbilt, from the Pohang University of Science and Technology in Korea, California State University, and Vanderbilt. And the authors, there are two co-first authors, Yu and Sheely, and then Ziba, Torres, Baltagolova, Thomas, Shelton, McGovern, Fudeding, Olson, and the senior author is Mariana Bindloss. She is an assistant professor at the Department of Pathology, Microbiology, and Immunology at the Vanderbilt University. And I want to highlight that in 2022, she was named in the first cohort of early career scholars of the Howard Hughes Medical Institute as a Freeman Harbalsky Scholar. And these people have been selected not only because they're superior science, but also they have demonstrated potential to really advance diversifying academic science and science in general. So delighted to highlight her work. So as Michael said, the topic is Salmonella typhimurium expansion in the flamed gut depends on aspartate derived from a reactive oxygen species mediated microbiota lysis. So as many of our listeners will be aware, we are endowed with many microbes. More than 1,000 different species live in our gut, and they provide a lot of benefits. They provide essential nutrients by digesting dietary fibers like cellulose. They also make vitamin K, and because they're consuming a lot of nutrients that, of course, are flowing through our GI tract, they also represent a formidable barrier for any interloper that wants to come in and try to gain a foothold, and it has to compete for nutrients. So their really tight food chain in the healthy gut makes it difficult for other microbes to gain a foothold. And yet, Salmonella typhimurium is going to be familiar to our listeners. In the U.S., it causes more than 1.3 million infections per year, more than 25,000 hospitalizations, and more than 400 deaths. And the people that don't die maybe wish they did, because they get diarrhea, stomach cramps, and fever that can last for days. And where do you get this? Outbreaks are one thing that make the news. Backyard chicken coops, as more and more people are raising their own poultry, is one classic source. And then in 2024, there was an outbreak that was traced back to basil that is packaged and sold, unfortunately, at Trader Joe's and other stores in 29 states. And then in TWiM 309, you may recall, we talked about charcuterie packs that had Salmonella contamination. And this is also why we're taught as kids not to eat the cookie dough, because there might be contamination from the raw eggs. So how is it that Salmonella typhimurium can overcome this, what's termed colonization resistance, this formidable barrier of the happy network of food chain that's in our healthy gut microbiome? So the authors tackle this question by applying a really impressive profile of bacterial genetics, mouse genetics, nutrient supplements, inhibitors, metabolic inhibitors, and germ-free mice to identify the key players that equip Salmonella typhimurium to gain a foothold and overcome this barrier. So to start, they knew from the literature that Salmonella benefits from living in the inflamed gut. And this actually builds on a groundbreaking paper from Andreas Baumer and colleagues in 2011. So other contributors were Sebastian Winter, Renee Solis, John Roth, and several others. And this is actually where our senior author now, Assistant Professor Binloss, trained as a postdoc. But they demonstrated that inflammation in the gut actually favors Salmonella, which can feed off nutrients, including ethanolamine, released by the inflamed intestinal cells, and use that as a food source. So this is really building off that and looking in particular at the capacity of Salmonella to use aspartate as a feeder into a anaerobic respiration pathway that uses fumarate as a terminal electron acceptor. So recall that respiration is a pathway in our cells where we can use carbon sources to generate ATP through a multi-step reaction called respiration. And part of that respiration chain involves passing waste electrons from one component to another. And then you need a terminal electron acceptor. So for organisms that live in oxygen or live in aerobic environments, oxygen is typically the final electron acceptor. But for all the microbes that live in our gut, where there's not a lot of oxygen, they can use other organic compounds as terminal electron acceptors. And in particular, fumarate is what we'll be discussing here. And aspartate can be converted to fumarate by an enzyme called ASP-A, aspartate ammonium lyase. So these are the key players that we're going to talk about today: aspartate, fumarate, this respiration chain in anaerobic organisms, and ASP-A in particular is a key enzyme. So what they wanted to understand is how is it's known that both E. coli and Salmonella have encode this ASP-A enzyme that can generate fumarate, and yet somehow Salmonella is gaining a foothold in the presence of all these enteropathogen or enterobacteriaceae that live happily in our gut. So that's the challenge they took on. So they started by demonstrating that indeed, when the mouse gut is inflamed, either experimentally by adding dextrane sodium sulfite, a chemical that triggers inflammation, or by adding pathogenic Salmonella typhimurium, sure enough, the amount of aspartate in the feces does increase. They also then tested, can Salmonella grow on aspartate? And they found that it could, as long as you also provided a carbon source, in this case glycerol. So Salmonella can grow under those conditions, and they require the aspartate enzyme to do so. But they do need the glycerol, so that allowed them to deduce that the aspartate was actually not acting as an amino acid food source, but rather a component of this anaerobic respiration chain. So they, having demonstrated that in vitro, they then went into the mouse gut and asked the same question: does the aspartate gene confer a fitness advantage to Salmonella? And when they force-fed the Salmonella that either encoded or didn't encode aspartate, and then looked out for as long as a week or 10 days, what they found is there was a fitness advantage, but it wasn't evident early on. It did appear after about 7 days or a week or so. So definitely aspartate was conferring a fitness advantage, but it took some time. So based on all their knowledge of Salmonella pathogenesis, they appreciated that it does take a while for the gut inflammation to occur. So their first thought was, is it the inflammation that is responsible for conferring this benefit to the aspartate activity? So to test that, they took advantage of Salmonella mutants that lacked their key virulence factor, which is called a type 6 secretion system. It's basically a needle-like projection that the Salmonella can use to deliver a whole panel of virulence factors directly into host cells. And one of the consequences is you get a host inflammatory response. So when they did the same experiment with Salmonella that could not induce inflammation, now they no longer saw that the aspartate converted a fitness advantage. So this allowed them to say, okay, we need inflammation and we need aspartate, and that confers a fitness advantage to Salmonella in the mouse gut. So next, they wanted to understand whether the aspartate was acting simply as a food source. And so what they did is they fed mice aspartate in the drinking water. And sure enough, that was enough to level the playing field. Salmonella no longer needed the type 6 secretion system to trigger inflammation. If they were artificially provided aspartate, then they did have a growth advantage. And it depended on the aspartate process. So now they're wondering, what else does Salmonella require to take advantage of this aspartate that's generated by the host? So they, again, applied a really nice panel of mutants. They found that chemotaxis was important so that Salmonella could swim toward the food source. They also needed a transporter so that they could bring that aspartate into the cell. And they also needed a locus that encoded a fumarate reductase enzyme, consistent with their model that Salmonella in the inflamed gut uses fumarate as a terminal electron acceptor. So they then wanted to ask whether this inflammation would benefit in mice as well. And so they, again, just did some verification experiments and showed that using a mouse experimental model, they in fact did need the FRD locus to generate the fumarate to support this anaerobic respiration. So next, they, having convinced themselves that aspartate fumarate pathway was conferring a fitness advantage to Salmonella in the flame gut, they wanted to know what's the source of this aspartate. So they repeated the experiment now, but they either supplemented the mouse diet with aspartate or they did not. And what they found is that the aspartate gene that allows them to respire on aspartate increased fitness whether or not aspartate was present in the diet. So
that's a very cool result.
Yeah, just such elegant experiments. The question comes up and then they design just an elegant experiment to test it directly, right?
It's so perfect. Genetics are always great.
Yeah. And then also to, supplement the diet with aspartate. No, we can't bypass the defect. So then they thought, okay, it's not coming from the food, but we know there are gazillion microbes in there. Maybe it's coming from other microbes. They're releasing it. So now they repeat the mouse experiment, but they use mice that have been raised to have no microbes in their gut. So these are called germ-free, which now that microbiology has advanced so much and we appreciate that microbes are so beneficial in our gut, I wonder if we're going to lose this term germ-free. Germ has kind of a negative connotation. But
We're going to go to notobiotic.
Yeah, notobiotic is the other term. So
right, and this is really cool that you raise these mice in a bubble. So they really are bacteria, archaea. They're free of every microbe free, maybe is a better term.
Yeah. And by the way,
they have a few. They have a few.
But they're not, their gut's not normal. The blood supply in their gut, the nerve supply in their gut is messed up. Architecture is different. So the microbes really are contributing to development. Yeah. But so when they repeat this experiment and now force feed the Salmonella into the mice that don't have a gut microbiome, they no longer see a fitness advantage conferred by this pathway. So the ASP-A mutant looks just the same as the wild type. So now they've got to come up with another source. What could it be? And they're thinking, we know inflammation is important, but commensal microbes aren't doing it. So what if we first colonize the germ-free mice with some beneficial microbes, which they do. They use some Bacteroides and Clostridia species, which are well known to be members of our normal microbiota. And in that case, now the germ-free mice, endowed with now their own microbiota, now, in fact, they do see that the ASP-A enzyme does confer a fitness advantage to Salmonella typhimurium, and that's shown in Figure 5. And likewise, they also, to set up their next experiment, show that they could just add one particular Bacteroides species, best known as B theta, and that too is sufficient to confer an advantage to Salmonella typhimurium that encode this ASP-A enzyme. So their motivation for doing some experiments and just proving that B theta can also confer this fitness advantage to Salmonella means they can now take advantage of genetic engineering tricks for B theta to do an even more clever experiment. Again, they're trying to understand where is this aspartate coming from. So they have an engineered strain that encodes a couple of cytotoxins that are sufficient to lyse B theta, but only when they're expressed. And it's designed such that if they add a particular chemical supplement to the diet, they can determine when B theta will turn on these cytotoxins and lyse, and therefore, spill their guts into the gut. So now they colonize germ-free mice with this, ticking time bomb B theta strain, infect with Salmonella that don't even have their type 6 secretion system, so they can't turn on inflammation. And the Salmonella strain either encodes this ASP-A enzyme or it doesn't. And then they feed the mice the inducer to trigger the lysis. And they do see that there is a modest decrease in the colony-forming units of B theta in the gut. So this experimental system is working for them. So then they were able to say, okay, does Salmonella that encodes B theta have an advantage now when we intentionally lyse B theta? And yes, it does. So this is pretty easy to picture. They've set it up so that B theta lyses, and now the ASP-A pathway is conferring a fitness advantage in the gut of these mice.
It's just like clever experiment after clever experiment.
Really clever. And I think the next one, I did skip over some because this paper is just full of just really elegant in vitro and then in mouse model experiments. But their final experiment then is this conundrum. Yes, we're convinced now that the ASP-A conversion of aspartate to fumarate is conferring a fitness advantage. But a lot of Enterobacteriaceae that are commensals in the gut also have the ASP-A pathway. So what gives? They want to ask head-to-head, is there something special about Salmonella typhimurium that distinguishes it from E. coli? So they do an in vitro experiment, and they find that no, E. coli actually isn't benefiting from aspartate the way that Salmonella is. And that's shown in figure 7. But they do show that both in vitro and in mice, Salmonella still require the asp-A activity. So they still require the ability to convert the aspartate to the fumarate. And then they see a really convincing competitive advantage of the Salmonella over E. coli that also have the asp-A pathway. And again, they don't see that advantage till about 10 days, consistent with the idea that it's host inflammation that gets triggered by the Salmonella type 6 secretion pathway, triggers an inflammatory reaction. The neutrophils and other mediators come in, release reactive oxygen species. That's sufficient to lyse a number of the commensals, which then spill out their guts, including aspartate, which Salmonella then can swim to through chemoataxis, take up through their transporters, and then use in this anaerobic respiration pathway to generate fumarate to act as their terminal electron acceptor. And also, it's clear that nitrate is also playing a role, and they didn't investigate that in a whole lot of detail, but it's also likely an even more efficient electron acceptor. So that is how Salmonella can make the best of a bad situation when it triggers host inflammation and reactive oxygen species are spilling out, killing the commensals, spill out their aspartate, and then Salmonella scavenges on that and uses it to generate ATP and bloom in the gut.
It's absolutely incredible. They are unleashing their own dogs from hell.
Yeah, it is also incredible because there's a lot of talk, right, in pathogenesis, at least when I 15, 20 years ago, about how bacteria sense when they're inside the host, but like dead other bacteria is not a bad way to do it, right?
Yes.
Especially ones like Bacteroides, which are only there.
Yeah.
I wanted to say, well, first of all, this paper is co-authored by two people, a postdoc, Woon Jae Yu, who we weren't able to actually end up contacting, and a graduate student, Nicholas Shealy. And Nicholas, actually, he talks about this, but he went to College of Charleston, and then he did a post-bac and an NIGMS post-baccalaureate research education program, or PrEP, which are great, at Medical University of South Carolina.
All right.
Cool.
Yeah, exactly. I thought that was really neat.
Our director, Laura Kasman, will be so happy we mentioned it.
Exactly. I was thinking someone would be happy, and not just Michael is even more. But Nicholas is originally from Columbia, South Carolina. He's a German-American biracial scientist, and he's passionate about making the scientific enterprise a place for anyone. He wants to be a PI someday because he really is fascinated by how gut commensals deal with life stresses, even if they don't have to worry about prom or taking the SAT. They're influenced by our diets and medication and lifestyle choices. And as a kid, he loved Animal Planet and the Discovery Channel. He originally wanted to be an MD, but he worked in Mark Harmon's lab at MUSC on bioactive secondary metabolites. And there he had to follow his gut-to-gut microbes, he says.
And metabolism.
Exactly. He wants to shout out Andy Goodman, who's a researcher at Yale, and Lynn Bentley, who designed the suicides construct that they used to kill the Bacteroides theta iota micron, which is the gut commensal that died and gave up its aspartate so Salmonella could thrive. And that was really instrumental for this paper. His advice, he's still struggling heavily with imposter syndrome. And I'm sorry to hear that. I feel like a lot of us, even at later stages, struggle with it. And you really just have to, he says, guard against comparing yourself to other people. And I think that's true in life in general, but it's very true in science because, each of us is contributing in our own way and our own thing. And you really can't compare and you can't count papers exactly or things like that. So I think that's great advice as long as you're interested and doing work you find rewarding and exciting and you're being paid to do it, ideally, I think that's what you should do.
And if I could just expand on that, I think it's a common experience that we compare ourselves to the people that are like 3 to 5 years ahead of us in our training.
He
which,
even mentions that he's comparing himself to his PI, which you don't need to
crazy.
compare yourself to your PI.
So Nicholas and all of our listeners, I'll just remind you of the old joke about the two hikers who are suddenly confronted with let's say it's a jaguar. Like, I don't have to outrun the jaguar. I just got to outrun this chump next to me.
That's right.
Exactly.
So
and
you don't have to outrun the PI, or you just got to outrun, stay even or ahead of your cohort in your training.
Exactly. And his 9 scientific advice is to laugh a little. Life is too short not to laugh. And his COVID hobby is knitting. And he's made a lot of scarves, especially in winter. I imagine it's not so fun to knit scarves in South Carolina,
no,
or Nashville in the summer.
Right.
He's into painting animal life with acrylics, playing D&D, and Magic the Gathering with friends. And he says he's sorry he's a little bit of a nerd. It's like we
don't apologize.
No,
you're on the perfect podcast, Nicholas.
Yes.
So I thought that was really wonderful. And Mariana, unfortunately, Wong Jae Yu, who's the first in the order on this, is now he couldn't respond. He's now an assistant professor at Post-Tech University in South Korea. And the PI emailed me and wanted to make sure that we acknowledged him too, even though we weren't able to contact him in the end. And she herself, as Michele pointed out, is very impressive. She even wrote a little bit about herself, but she is a DVM, a doctor of veterinary medicine, and a PhD from Universidade Federal de Minas Gerais in Brazil. And she's originally Brazilian. And she decided to become a scientist during high school after learning about genetics, evolution, and physiology and biology. And she was inspired by her middle sister, who was going to vet school.
Wow.
And so that's kind of where she got in. And she became fascinated by bacteria in vet school. And I think that's what led her on this path. So, that's a slightly different path. Usually people decide, before they go into professional school, which professional school they go to. But I think it's great that even, she made her decision a little differently. But in the end, this is really beautiful work. And I hope she's super proud of it because it's really cool paper.
Mm-hmm.
And so all the authors should be on this paper.
Exactly.
And again, folks, this was an open access manuscript.
Yeah.
So
and I'll
thank
just
you,
say
Michele.
one more thing about the, habits and are we nerds or what? But that's really what drew me to science. My first job as a technician, I just was surrounded by people from all different walks of life, all different backgrounds, very wide range of personalities. But yet, they were all smart and they really cared deeply about what they were doing, loved to think and try to solve these riddles. And I just loved being in that milieu, that environment. So, come one, come all. It's a great place to spend your careers.
Your life.
Yeah.
Well, thank you, Michele and Petra. But before we close, I'd like to share some sad news with you. I just learned that one of the giants of our discipline and our longtime volunteer who supported the American Society for Microbiology has passed. And that individual was Dr. Sam Kaplan. To those who knew him, appreciate he was larger than life. He was for many years a force in scientific publishing, especially for ASM, as the editor-in-chief of the Society's flagship journal for the years he was its director, the Journal of Bacteriology, whereas Stan Malloy quipped, if you reviewed a paper quickly and well, another review was quickly added to your queue. Dr. Kaplan was also the treasurer for ASM, where he and his finance committee well guided the society and invested its limited resources so that ASM would remain financially healthy long after his term as treasurer ended, where he was a longtime champion of ASM branches, but especially students and postdocs, especially the undergraduates, and their ability to attend our annual meeting, where he was instrumental in ensuring that these future scientists could experience their first large gathering of like-minded nerds, if you will, as well as ensuring that our branches had resources to nurture and encourage that next generation of microbiologists through the support of their local meetings. Additionally, Dr. Kaplan held faculty appointments at 2 microbiology powerhouses. First at the University of Illinois, Urbana-Champaign, where he was a colleague with other giants of our field like Erwin Gonzalez, Carl Woese, Abigail Saylors, Dixie Witt, and one of our founding TWiM hosts, Stan Malloy,
Yes.
just to mention a few. He left Urbana in 1989, and he then became the founding chair of the Department of Microbiology and Molecular Genetics at the McGovern School of Medicine at the University of Texas in Houston. And upon the 30th anniversary of his beloved department, they held an anniversary symposium in his honor in 2019. Finally, in closing, no tribute to Sam would be complete without mentioning his long and productive research career, where he and his countless graduate students and postdocs principally focused on the life and times of the purple photosynthetic bacteria. I was fortunate to have interacted with Sam over the years through our mutual volunteer activities for ASM, and I can personally attest there was no better friend to microbiology than Sam Kaplan. So he was a force.
Michael, thank you.
Thank you, Michael.
Well, we hope you enjoy today's science offerings. Show notes are available at microbe.tv/TWiM. If you like our work, please consider supporting us financially at microbe.tv contribute. Any amount is always greatly appreciated. It helps us in our ability to support continuing to bring you such cool science. I should also mention that URL, microbe.tv, will help you locate the microbe.tv store, where you can purchase all sorts of merch, where $1 from each item purchased comes back to help us pay the subscription to keep the podcast up on the servers to help support our work. Your contributions, if you're in the U.S. to Microbe TB, are tax deductible if you're a U.S. taxpayer, as the U.S. IRS deems Microbe TV as it is a 501c3 nonprofit entity. Further, we'd love to get your questions and comments. Again, just write to us at twim@microbe.tv. And Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you. My pleasure.
And Petra Levin is at the University of Washington. Thank you, Petra.
Actually, Washington University.
Oh, I screwed it up, and it's written down.
It's okay. It happens, but very much in Missouri. Anyways, thank you so much, Michael, for being our fearless leader today.
And I'm Michael Schmidt from the Medical University of South Carolina, filling in for our beloved podfather, Vince Racaniello. Hope you enjoy your dinner or the podcast tonight. I'd like to thank ASM for their support of TWiM, Ronald Jenkins for the music, and of course, Ray Ortega for editing of this podcast. Thanks for listening, everyone. See you next time on This Week in Microbiology.